All right, I am going to record. I am recording now. So you will be, if you have your camera on, I do have to upload them to YouTube. It's just to my school YouTube channel. I don't tag it with anything. So like nobody should really be finding it. But it's the only way for me to get it onto Schoology because the file size is going to be so big for the kiddos that can't log in. So just for your awareness. So yeah, okay. What questions do you have?
I can start.
Okay.
Let's go, Eddie. Okay. What exactly is CDK?
CDK?
Like
Okay, so CDK stands for cyclin dependent kinase. And I'll type that in the chat.
Yes.
All right, so it's a cyclin dependent kinase. So we know it's an enzyme because he ends in ASE, right? And he is one of the enzymes that will stop the cell cycle if something is wrong. And it actually has to do with like the amount of cyclin that is produced. You don't need to know any of those kind of levels of details. But basically, we know that in the cell cycle, as it goes around, there are checkpoints, right? Where the cell will have something to tell it, okay, everything looks good and you can go, or no. So things look bad and we need to stop. And basically, CDK or cyclin dependent kinases are one of those enzymes that put the brakes on the cell cycle. And so you can have cancer if your cyclin-dependent kinase is not working properly. So think about it if it was a car, okay? So CDK is like brakes on a car. So if you're driving your car and you hit the brakes and they don't work, what's going to happen to your car? Your car is going to keep going. So instead of stopping the cell cycle, it would keep it going, and then you would have a proliferation of really bad cells. Okay. Good question.
Okay, my question kind of goes along with that, but is that gonna be an oncogene?
So oncogenes are gas pedal genes. Okay, so CDKs are like the brakes, and oncogenes are the gas pedal genes. And so if everything's going well, you want your gas pedal to go, right? But sometimes your gas pedal could be stuck down, and that would be how an oncogene would result in cancer. And we've named them oncogenes because that defect of the gas pedal being stuck down is more common than your brake pedal not working. Okay. And so we have cancer as a result of oncogenes not working more often than we do with the CDKs not working.
Okay, I was also gonna ask if the G1, G2, and M checkpoints happened at the same time as like G1 and G2 phases.
Yeah, and that's how they get their names. So it's because of the cycle, the phase that they're in, and it happens towards the end of that phase. So if it's a G2 phase checkpoint or a G2 checkpoint, it's going to happen at the end of the G2 phase. So that one's going to be looking for replication, if there's enough space in the cell, if there's enough of the organelles, like do we have 2 copies of the centrioles so that we have 2 different centrosomes. Emma is here.
Hi, Emma.
Hi, Miss Emma.
Hi.
You haven't missed much. We've just been talking about the cyclin-dependent kinases and oncogenes, okay? And just how they are different enzymes that work for the checkpoints to make sure that the cell can or cannot progress through to the next phase.
Okay, what did you say that they were again? Sorry, my mom.
No, you're fine. So it's the CDK or cyclin dependent kinase. I dropped that full name in the, oh, you can't see that. Let me drop it again in the chat. I did learn if you join late, you can't see previous chat things. And then oncogenes, I'll drop that in the chat too. So both of those work as the checkpoints. CDKs or cyclin-dependent kinases work as brake pedals, just like in your car, where they would stop the cell from going through to the next cycle. And oncogenes would say, yes, yes, you're good, you can go on. And if your brakes or your CDKs are not working, then you could go into the cell cycle incorrectly and make cells that are not good, which could lead to cancer. And then the same thing with your oncogenes, your gas pedal could be stuck down and you could go into the phase faster. So you could be making more cells at a faster rate than you should be.
Okay, thank you.
Yeah, no problem. And I am recording everything too. So if anybody wants to go back and listen, if you feel like you've got it, but then after we get off, you're like, oh my God, wait a minute, I don't got that anymore. Take another listen. It'll probably take me about 20 minutes to upload it to YouTube and then drop the URL. And then if you're still confused, message me. What other questions do you got?
Does the G2 do?
G2. So G2
Sorry, Laura.
phase
No,
or
you're
the
good.
G2 checkpoint?
The phase.
The phase. Okay, so in the G2 phase, in G1, we've grown bigger as a cell, right? And in G2, we're basically making any enzymes or organelles that we need to split into two different cells. Okay, so we're going to be making more microtubule pieces that can form into spindle fibers. We're going to be making another set for our centrosome. We're going to be making more mitochondria and ERs and Golgis, all of those organelles that will need to go into the new cells. So it's a building phase, but not building for the cell membrane, building for the internal components.
Okay, cool. Thank you.
Yeah, no problem. Miss Anna Laura, you were trying to speak, but Hayden beat you to the punch.
Okay, I think the biggest thing that I struggle with is just like the cellular respiration thing. So I was gonna ask you if you could explain link reaction, and also like I wrote down the complexes of the electron transport chain, but I don't really understand what I wrote.
Okay.
Because I wrote it like in textbook language.
Yeah, that's not a fun language. Not at
Yeah.
all. All right, so I think I have my phone here so that I can try and write things. I'm going to try and be really fancy. All right, screen mirror, come on, there we go. Why are you not clicking? There we go. Okay, and then everything seems to be working very slow for me today, which is not fun. All right, here we go. All right, so this is my fancy at-home document camera. It is my phone on a glass. It's great. All right, so the link reaction. What do we start with? Well, actually, let's back up a little bit. What process has just occurred in the cell?
Glycolysis.
Glycolysis. Awesome. So it happens after glycolysis. And what did we make in glycolysis?
2 ATPs and 2 pyruvates.
Yes, so 2 pyruvates or pyruvic acid. So as long as you can recognize those two as being the same thing, I don't care if you have to write what you call them, okay? So pyruvates. So we have a pyruvate and we need to get to the citric acid cycle. And in order to start the citric acid cycle, what do we need the pyruvate to be?
Acetylcholine.
Yes, acetyl-CoA.
Oh, choline. Okay.
Yes, CoA. All right, so acetyl-CoA. So pyruvate has 3 different carbons. Carbon, carbon, carbon. It has some hydrogens and oxygens associated with it too, but the big takeaway is that it is 3 carbon molecules. And acetyl-CoA has 2 carbon molecules with this weird coenzyme A attached to it. So we need to do some stuff to get from 3 carbons to 2 carbons. What do we need to do? What's an obvious thing that we might need to do?
Get rid of one of the carbons.
Yeah, we need to get rid of one of the carbons. And what is that called when we get rid of a carbon?
Oxidation.
Decarbonization.
Yeah, almost. Decarboxylation.
Decarboxylation.
Oxylation.
Wait, why wouldn't it be called oxidation? Because isn't that losing another element?
Yes, but remember oxidation, and let's come down here just a little bit. Oxidation is losing electrons. Okay, so we may be doing decarboxylation and oxidation together. Okay, but the first thing we do know is that we need to get rid of a carbon, but that carbon's not going to be by itself. What is he going to go off to become? He's got to go and float off into the atmosphere.
I don't know.
It's all right. It's all right.
Does it turn into carbon dioxide?
Yes, carbon dioxide. Okay, so that one little carbon is going to be carbon dioxide and he's going to float into the atmosphere. But when we've done that, we've also, as Callie mentioned, we've lost some electrons. Okay, so it's going to be a decarboxylation and an oxidation reaction together. And who's going to pick up our electrons? So we're going to have 2 electrons that we've lost.
Isn't that the electron transport? Or is that not?
So we're going to make something that's going to go to the electron transport chain.
ATP.
Not ATP. That's a final product.
NA.
Yes. Now it's NAD. What? Because we're in cellular respiration.
B.
Is it NADPH or is it just NADH?
H.
Just H. Okay. So we're going to make NADH. Okay. And then we need to add this weird little CoA. Okay. Coenzyme A, he is a derivative from a vitamin. You don't need to know that. And so he's just going to come into the reaction and add to the last 2 carbons. So that's really all that there is to the link reaction.
So it's like carbon dioxide and NADH are like products or like waste products?
Well, carbon dioxide is waste. So this is waste. But this is not waste. Where does this go now that it's been made? Where can it go?
Won't it go in like the Krebs cycle?
Not the Krebs. So it doesn't go into the Krebs cycle, but we make more in the Krebs cycle. What comes after the Krebs cycle?
The electron transport.
Yeah, so it can now go to the electron transport chain to help make more ATP.
Okay, I also have a question about the electron transport chain because I watched one of the videos. Well, I want to do the complexes, but like also, is NAD plus just like a carrier?
Yes, yes. Remember, let me move down my fancy document camera, that in order to make NADH, okay, this goes either direction, we would need NAD plus 2 electrons plus a hydrogen ion. Okay, so when we're making NADH, we would take these 4 things and add them together. And then when we get to the electron transport chain, we take this guy and we break him into these 4 things.
Mm-hmm.
Do we just need to like know and like be able to like recognize that that's happening, or would it just like that? I don't know, all of these like things are changing in other things, or is it like going to be like a short answer question where like we have to answer it and like figure it out?
No, your short answer are a little bit more basic in general, or something we specifically talked about. Not that much detail. You will need to know this kind of stuff for your multiple choice, but not necessarily for your short answer.
Yeah, and so like stuff with like the Calvin cycles and stuff, do we need to be able to like draw those and like know like just know what's happening, or do we need to be able to like do what's happening? You know what I mean?
If you can draw it and understand what's happening, that's like A+ material. And as far as your understanding, you're not going to need to go into that much depth on a short answer or a lab-based question. Those would just being able to draw and understand all those details is going to help you with your multiple choice questions.
And will we have 4 lab questions this time?
No, you're only going to have 3 because remember that one lab failed.
Oh, right.
Right. So I just took it off. Yeah.
Okay.
Okay, so oxidation happens in the link reaction when we take and get CO2 out and NADH, right?
Mm-hmm.
Okay, but more specifically from the NADH because we end up with the 2 electrons, right?
Yes.
Okay. So then how does the oxidation happen in glycolysis?
Okay, so remember that oxidation means we are losing electrons. So anytime you have a molecule that's losing electrons, you're going to have oxidation. And so where it happens is when we go from glucose in glycolysis, let me write over here, this is glycolysis. So when we go from glucose to our first big molecule, we've added 2 ATPs, right? So we've phosphorylated that glucose, 1, 2, 3, 4, 5, 6 with a P and with a P. Then this guy here is going to get split into 2 different pieces. And remember, this is where I was like, you don't need to know their names. This guy will then get oxidized because he will lose electrons and form NADH. Both guys will. So remember that, I ran out of room. These guys are getting oxidized, but the NAD plus is getting reduced. So it's actually called a redox reaction because you have whenever you have one molecule that's getting oxidized, you have one that's getting reduced. But we also focus on this part being an oxidative reaction because we're focusing on what's happening here to these main culprits, these main guys. Okay, and then this is where we end up with the pyruvates. So anytime you have one of your main molecules losing electrons, which means typically that either NADH or FADH2 is produced, then you have had an oxidative reaction.
Okay.
You said the link reaction is preparing for the Krebs cycle, right?
Yes, that is correct. And when you're studying, don't forget to be looking at your learning objectives. Some of your learning objectives for these topics are really general. So remember to focus on those so that when you feel like you're getting lost in the weeds of the details. Because there's a lot of details, but you're not necessarily responsible for all the nitty-gritty details.
Some of the learning objectives
Mm-hmm.
I cannot see. I'm going into Schoology to see exactly.
Yes,
which ones
please,
those
so
are.
I can fix that because that's not okay.
Okay, I can't see chapter 10.
Can't see chapter 10. Okay. Let me fix that.
And also chapter 11.
What? I'm a bad teacher. Bad. Well, I'm glad I brought them up for you guys to look at so that you guys could tell me what you can't access. All right. It takes me just a little bit to get there. I'm going to do this now while we're talking about it because I don't know about y'all, but if I don't do it right now, I'm going to forget. All right, but you can get into 7 and 8 fine.
I
Probably
can
double check
get
those.
into
Just to make
8
Yeah.
and double check 7.
All right, learning intentions.
Yeah, all the other ones are good.
Okay. Yep, look at that. It says restricted. That's not cool. All right. So I need to pause share maybe. Do something to move it so that I can access this. It's like blocking right where I need to click. Oh, my life. Fun times. Fun times. All right, here we go. All right. That was chapter 10. Took me that long. Just do chapter 10. All right, let me do chapter 11 real quick. Then I need to remember to go and fix those in the future when I'm making the new ones. All right, what questions do you guys have? I'm almost finished with chapter 11. I think I can handle a question if you've got one.
Do we need to know? Well, I guess the learning objective could tell me, but
That's all right, but I'm just opening them, so and I'm here.
specific suppressors like P53 and stuff.
Nope.
Okay.
Nope.
Just like what they do.
Yep, just in general, that basically that whole analogy of like the brake pedal and the gas and that they're checking for errors.
Okay.
All right, chapter 11 is officially fixed.
Is crossing over good or bad?
Crossing over is great for the most part. So it really just depends on the genes that the parents have. Crossing over leads to more chances of variety, which we like if we're humans because that makes us different than our siblings. So we love that. And then in the wild, in every other species, we like it because it can lead to variations that can improve our chances to survive in the environment.
Okay.
But if we have a jacked up gene, then crossing over, could increase the chances of passing on the jacked up gene.
Wait, okay, I can't remember which one it is, but I'm gonna try my best.
Yeah,
but like
absolutely.
mitosis prophase. Okay, in mitosis prophase, that's not when the nucleus goes away, but in prophase 1 of meiosis, the nucleus goes away.
In all prophases, mitosis prophase, prophase 1, and prophase 2 of meiosis, the nucleus disappears.
Okay, and that's like basically all that happens, right? Or like crossing over can also happen in
Right.
meiosis.
Right. So you have a lot of things that's happening there. It's a lot of like organization stuff that's happening. So the big thing is that your nucleus disappears, but also that your chromosomes are condensing. They're switching from the spaghetti, the yarn piece, into the bulky chromosome that will be part of the fluffy X. So that all of that's happening. And then your centrosomes are starting to migrate to opposite sides. Okay.
Okay.
And so that will start at the beginning of prophase and it will be completed by the end of prophase. And that is one of the things that takes the most time, is that migration of those centrosomes. And then you have your spindle fibers being built. So all your little microtubule pieces attached to the centrosomes. Those spindle fibers are also attaching to the centromeres, the connectors in the chromosomes. So lots of little things just to make sure that everything's gonna happen well.
Are we gonna need to know like the individual structures of photosynthesis?
No.
Okay.
You really just need to understand how the structure of ATP synthase works. And that's the same whether you're in photosynthesis or cellular respiration. Okay. The fact that you need a hydrogen ion buildup in a small space. So in photosynthesis, it would be the lumen of the thylakoid. Here, let me draw it. I forgot that I had my document camera. So in photosynthesis, you would have your chloroplast with your little thylakoids. Oh, you can't see it. I'm off to the side. Thank you. All right. So this inside space, the lumen, would be where.
I can't see it. It's like frozen.
There we go.
Okay.
Yeah.
There we go.
Thank you. Thank you.
Wait, so what is this? Sorry.
No, it's fine. All right. So here is our chloroplast. Chloroplast. This is technically, it's a stack of thylakoids. So what is that called?
Grana.
Yes, grana. And so within this individual thylakoid is a lumen on the inside. Lumen just means like hole or opening. And so when we get to our anatomy section in the spring, we're going to have lumen all over the place, okay? Almost everything that we look at is going to have a lumen. So just the inside of the thylakoid is a very tiny space. That very tiny space is essential for the hydrogen ions to build up to power that ATP synthase. And then when you look at your mitochondria, if I can get them both in here. Excellent drawing, I know. All right, so this is your intermembrane. So inter means between space. And that space is also very mitochondria. That space is also very small, which is important in a structure to function relationship for that ATP synthase to work. You need a small space that can quickly accumulate a proton gradient with hydrogen ions that will power that ATP synthase. And then the other, I'm sorry.
No, go, sorry.
No, it's all right. And then the other structure that you really need to understand how it works is the photosystems. Photosystem 2, which comes first before photosystem 1. And those are the guys where the chlorophyll pigments and all of your other pigments, if you are a plant that has more than one pigment, more than one color that you can be shown, will live in those photosystems. And those photosystems capture the energy from the light because of those pigments, and then the pigments can transfer that energy to electrons.
The things you just drew, the mitochondria and the chloroplast, are these? Are you talking about like the difference between cellular respiration and photosynthesis?
Yes, how the structure of the chloroplast, the thylakoid lumen, and the intermembrane space in the mitochondria, although they're different, they're still the same, and they have to be the same because both of these processes require ATP synthase in order to make the ATP, right? ATP synthase literally means ATP maker. And that's the whole point of cellular respiration, and it's an intermediary point of photosynthesis. So both of these organelles need to have very small structured spaces where that hydrogen ion concentration can build up into a gradient, and then they can diffuse through the ATP synthase, which will power it and allow it to make ATP.
And are the photosystems, okay, so that's like photosynthesis, and is the order of it is like photosystem 2, and then is that the proton pump that's in between?
Yes, yes. And you don't need to know really any of the names for the molecules in either of the electron transport chains. Just know that you have proteins within the membranes that move the electrons, hence the name electron transport chain. And as they move the electrons, they also shoot hydrogens. They pump, if you want to remember the idea, the name of a proton pump, they pump hydrogens to the other side. And that's where that buildup of hydrogens is coming from.
So would that be at the end of the electron chain transport, or the
That would,
electron?
yeah, that would be at the end of the electron trains. Well, kind of the end. So in cellular respiration, it is almost all of it. Like, so is the moving of the electrons and pumping of the hydrogens, and then we throw that ATP synthase on at the end. In cellular in photosynthesis, sorry, the electron transport chain is in the middle of the two photosystems. And then it's the electron transport, the ATP synthase. Here, I'll show you some pictures. All right, so move my fancy document camera up so I got a lot of space. All right, so we'll start with our membrane. I'll draw one little phospholipid on each side so you can see that it's a membrane. Oh, you can't see that. Here we go. And you have your photosystem blob. That's photosystem 2. And then you've got proton pump. You've got some weird little other things. And then you've got another photosystem. And another little guy. But you don't need to know these little guys or where they're located, okay? All you need to know is to get from here, photosystem 2, to photosystem 1, that this is called the ETC, the electron transport chain, okay? So this space here is your electron transport chain, and this is in photosynthesis.
That's really all we have to know?
Kind of, yeah.
Hank Green goes so in depth.
Oh, yeah. No, he does. And that's why be happy that you are in ACA Reynolds biology. Okay? This is one time where AP goes into a lot more detail than you guys do. Okay? And then this last guy is our ATP synthase. And then if you are one of those lovely people going on to college biology, have super fun. In biochemistry, you have to know all of these guys, not only what they do, but what their exact names are. Super fun.
It's terrifying.
It was almost the death of me, but I survived. And at that point in my life, I didn't know how much I liked biology. I didn't know how fascinating it was. Cellular respiration is very, very similar. Except for we've got unknown guy we don't need to know, but we do need to know that he breaks down the NADH into NAD plus. You've got another little guy that you don't need to know his name, but you need to know that he's the guy that does FADH2 into FAD. And then you have your ATP synthase. That one is so ugly. Oh my gosh. So yeah, that's your big picture right there.
Okay.
Oh, and then this, I'm sorry, then this whole thing here to here is our electron transport chain. Sorry.
Okay, and we need to know like the two parts of photosynthesis of like the light reactant, the light dependent and light independent reactant.
Yep, the light independent or Calvin cycle. You do need to be able to recognize it either way. Just like
Oh,
with
it's the same
citric
thing.
acid cycle and Krebs, it's the same. Okay.
Wait, what? Sorry, the light dependent is the same as the Calvin cycle?
The light independent.
Oh,
Independent reaction equals Calvin cycle. And there we go. The citric acid cycle equals the Krebs cycle.
oh, okay. That's,
And why don't they just call it the same thing?
yeah.
That's a little odd.
So Calvin and Krebs were the scientists that discovered it. So that's one of the reasons why we call them those names.
Oh.
Yeah, gotta get
Yeah,
on
but
there. I
it just
know.
makes it so much easier to know.
Yes.
Okay.
I prefer calling the Krebs cycle the citric acid cycle because citric acid is the first byproduct, and that's why we use that term. Light independent reaction, you actually hear the cycle being called the Calvin cycle more than anything else because of the misconception that we're starting to realize that students have in that light is not necessary.
Mhm.
And it's not for that cycle, but if you don't have light, then you're not going to have the cycle because the light makes the products that power the cycle. So some students are sometimes incorrectly think that the Calvin cycle happens all the time and it happens in the dark. It doesn't actually happen in the dark, it happens in the light. It just doesn't require light directly.
So with that, wait, hold on. Okay, so the light independent is the Calvin cycle.
Yes.
So what is the light dependent reaction?
This guy up here is the light dependent reaction. So that includes the electron transport chain. It also includes the splitting the water, the photolysis here at photosystem 2. And then we also up here at photosystem 1 at the end will make NADH.
Okay.
So we should know like the reactants and products of all these things.
You should know big reactants and products for all 5 of the subparts. Okay, so like what do you need to go into the light dependent reaction? Let's start
You
there.
need hydrogen?
Where do we get the hydrogen?
No, you get it from water.
Yeah, so we need water. Okay.
Okay.
And then what do we make from the light dependent reaction? Let's do this.
Well, aren't you making ATP?
Yes. What else? Light
Wait,
dependent.
I have a
Like
question.
this for light dependent reaction. Does that make sense?
Did you say that you need the water for the light dependent reaction or the light independent reaction?
Dependent.
Okay.
We're going to make a little list. All right, so we need water, reactants, products. And we just said we're making what?
ATP.
ATP. Okay, what else are we making?
Something probably
For, yeah.
AD pH? No.
Yes. How do I remember that I need the P here?
Because it's photosynthesis.
Because we're talking about photosynthesis. Awesome. And then when we split that water up, we're going to use the hydrogens and the electrons, but what's left from the water that we need to do something with?
Oxygen.
Oxygen. We're going to have oxygen gas released. All right, and then in the light, let's say Calvin cycle. That's easier. What are my reactants? And what is my product? So I'm going to use two things that I made over here in the light dependent reaction.
You're using the NADPH.
Yep.
And aren't you also using the ATP?
Yep.
Because those are the things that are like net yield, right?
Yes. Yes. And then we need something from our atmosphere.
Carbon dioxide.
Carbon dioxide. And then what are we making?
You're making more ATP and NADPH. Right?
No, we're breaking those down. We're actually going to make ADP and NADP plus. But those will just come back and get recycled back and forth. So I tend to leave those off of a big list where you're just looking at like your important characters.
This is still a plant though, right? So we're still
Yeah.
going to make oxygen then?
Well, we've released our oxygen up here in the light dependent reaction. All right, guys, you're getting stuck in the weeds.
Toast.
Yes.
That was so dumb, yeah.
That's the only thing that we make that we care about. Okay, so if you were to write your formula, okay, you would have water and carbon dioxide on your reactant side. You would not include NADPH or ATP typically because we make them in the light dependent reaction and then we use them immediately in the Calvin cycle. So they're important, but if we're writing our overall reaction, we wouldn't use them. And then we're going to make glucose and oxygen. Okay. All right, want to do the same thing for cellular respiration?
Yeah.
Yeah, see some nods of the heads. Yes, please. Yes. And I can scan this chicken scratch weird stuff to go on as well. All right, so let me line up the document.
The formula for this one is C6, just to like see if we do it that
Oh,
way.
yes.
6H12O6 plus 6O2.
Yeah, it's the
It's equal,
the-
or like arrow to the 6CO2 plus 6H2O.
Yes.
it's just like the reverse.
Yeah.
Light.
Yes. And we're making ATP. Oops. Oh, that was awful. Okay. Yes. Very, very good. Very good. All right. So the first step that we have.
Wait, I'm sorry. This is so
Yes.
stupid.
No, never.
Are they're both making ATP?
No.
Isn't ATP sugar though?
ATP is our cellular energy carrier or money. It's our
But
money.
like doesn't sugar make this, doesn't sugar give you energy? So aren't they like the same thing?
Sugar does give you energy in like that's what we think, but really it's not usable energy. Okay? So we need to take sugar, which has a lot of energy in it, but we need to make this ATP, this usable cellular currency. Kind of think about it as like glucose is your paycheck. Okay? That gives you money, right?
Mm-hmm.
But if you don't take it to the bank, you can't use that money.
Okay.
Right? If you don't deposit it into your account or cash it, that's not usable money. It's a beautiful check and you have it, but you can't do anything with it.
Okay.
Does that help?
Yes.
Okay.
So the ATP would be like the bank.
The ATP would be the cash you get from the bank or the money in your
Oh,
account.
okay, yeah,
The bank
okay.
itself would be cellular respiration.
Okay, got it.
So ATP is the ATM.
Yes. Well, it's the
Okay.
cash you get out of the ATM. Yes. And then you could think about cellular respiration being the ATM.
Okay.
Yeah. Okay. All right. Glycolysis, reactants, and products. What goes into glycolysis?
Glucose.
Glucose. Anything else?
Oxygen.
Nope.
Phosphate.
No
Right.
oxygen.
Oh, you
Would you
get a
phosphate?
phosphate out of it.
Yeah, so we get phosphates out of it. So what do we need to input?
Pyruv.
Not pyruvate.
Nope.
I don't really know.
Wait, I thought
Hmm?
Is it like a hydrogen or an electron or something?
It's actually one of the things that we were just talking about in our bank analogy.
ATP.
Yeah, we need to input some ATP. So glycolysis is like you're starting a business.
Because
Okay.
in glycolysis, you need two to do it, but you make
Yeah.
four, so that's why you yield two.
Exactly. Exactly. It's like maybe if you've ever had a job where you have to buy your uniform, you've got to upfront that cost, but then you're going to get your paycheck's going to be big enough to cover it. All right. So we're going to do 2 ATP. And that's all we need. So we're done with our reactants. So our products, what are our products?
4 ATP.
5
Yeah, we need 4 ATP. I'm just going to write that a little lower only so that it's across from my other ATP.
Pyruvates.
Pyruvate. How many pyruvates?
2.
Excellent.
And then did you say phosphorus?
No phosphorus.
No, it starts with the PH. No, what is it?
There's no P. There's no P because our only P is with our ATP.
Oh, okay.
Okay.
It
so
goes
we've
with
done that.
ATP.
Yep.
Okay.
But then we're making something else at the end, right before we make our pyruvates.
NADH.
Yes. Anybody know how many of those we make?
4.
1.
2.
2. It's 2.
Oh.
Yeah. 2. All right. We feel okay with that?
Yeah.
Okay. Let's go over to, I'm going to combine the link reaction and Calvin cycle, Krebs cycle. Well, listen to me together. Okay. All right. What do we start with the link reaction?
The pyruvates.
Yep. Okay.
And
Do we
then
need anything else to get this started?
Yeah.
Yeah, we need one weird little guy in the link reaction.
Your CoA.
Yep. Yes, your coenzyme A. OA. All right, what are we going to make for our products?
ADP.
Not ADP.
ATP.
Yep, we're going to make some ATP.
And acetyl-CoA.
We are going to make acetyl-CoA, and you can include that, but I wouldn't necessarily do that as an overall product because we're including this Krebs with it. Okay, does that make sense? Like if we were just focusing on the link, we did that earlier over here, right? You can say that you get your acetyl-CoA as the product. But for our big picture, which is what we're focusing on right now, I wouldn't include that one.
Okay, so would you also make NADH?
Yes.
And FADH?
2, yes.
Okay.
FAD has, when we join it, FAD by itself does not have a charge, so we have to include 2 hydrogen ions with that.
Okay.
All right, and we make one more thing, and this is a waste product.
Carbon
Carbon
dioxide.
dioxide.
Can we make 4?
Yes, so now you'd have to decide if you're listing your products here and identifying how many, are we going for 1 glucose or 1 pyruvate? Do you see? Because if you were doing 1 pyruvate,
Uh-huh.
then you would make,
Okay.
yeah, you would make 1 ATP,
Okay.
1 FADH2, and 3 NADHs and 3 CO2s. But if we did 2 pyruvates, because we were doing 1 glucose, then we would make 2 ATPs, 6 NADHs, 2 FADH2s, and 6 CO2s.
Okay.
Okay, so see how that depends number-wise on how many, you know, what your question says.
Is that going to be that specific on a question? Like, okay.
Could be. So really just with that, pay attention to detail and pause, okay?
Okay.
Just pay attention to detail and pause if you get a
Okay.
question like that. All right, we good over here because we got one more.
Yeah, the citric acid. No, we already did
you
that.
okay?
Just kidding.
The electron.
Love you, Emma.
Oh, like, I don't know. I just feel overwhelmed.
Yes. This test is awful.
I'm like,
oh,
I don't
great.
know. I'm just scared. I'm
Yeah.
just scared, Miss Sheely.
I think once you get your test, you're gonna be relieved.
Yeah,
Okay?
because
I think you're gonna be like, okay. Yeah, you guys are thinking like totally worst case, getting really stuck on the details, which tells me that you really care and you really want to be in it. But when you go back, and one of the first things I want you, first of all, I want you all to take a break from biology after this Zoom, okay? Yes, Emma.
I can't.
30 minutes, just 30 minutes, okay? Go and talk to your parents, walk around your house, stay off your phone though. Don't get on your phone. Take a brain break. You cannot overwork your brain, okay? There is a such thing as studying too hard. You don't want to do that. Take a brain break, and then the first thing I want you to do after your brain break is I want you to go into these learning objectives and start reviewing them, okay? Looking at them and seeing the level of detail that these learning objectives are asking. Because remember, the big thing is I'm not going to ask you anything that a learning objective does not ask you, okay? It can be, a lot of these details are within learning objectives, but some of these details are
Really
some of the ones that you guys are really, really concerned with, like we've already talked about, but they're not covered within the learning objectives, okay? They might be covered through the chapter, but there's too much detail, okay? All right, so our reactants and our products. All right, so what do we need to get the electron transport chain started?
The NADH, right?
Yes, we need NADH. What else do we need?
The FADH2.
Yes, ma'am. All right, and then any other reactants we might be needing? Go up to our formula up here. Have we used everything?
The H.
There's nothing by itself, so this is something. Yeah, we haven't used that oxygen yet.
No.
So we need our oxygen. Okay, what are we gonna make?
ATP.
ATP.
Hydrogen.
We're gonna make some hydrogen, but remember that hydrogen is just temporary. to
I
power
thought
the,
it accumulates.
huh?
I thought that accumulates.
It
and then-
does, but we're either going to shuttle it back
Mhm.
Krebs cycle so that it can be made here with NADH or FADH2, or it's going to become part of this product that we're missing,
Oh.
which is also up
Oh.
here.
H
Wa-
H-O-T.
Water.
Yes, water, H2O.
Did I really say HO2?
You did.
Oh my
Wasn't gonna
god.
figure it out. I know I was just a brain
That's
fumble.
what I meant.
Oh,
Okay.
it's a long Saturday. You definitely need a brain break, Callie. Definitely.
Okay.
It's okay.
Okay, and we made 30-ish ATP. right?
Yeah. Yep. For one
That-
glucose, you would make anywhere between 30 and 36 ATPs.
And does that number change if it's like, because these are in like humans, right?
Yep. Uh-huh.
Does that number change throughout animals, or is it like the same throughout all things?
Yeah, it's pretty efficient throughout all. The variation between the 30 and the 36 that we see is how efficient the FADH2 protein is in shuttling. For some reason, we see him being not consistently efficient. And you do not have to know this at all, but just if you're curious as to like, why is there range? Why is it not just a flat out number? That enzyme, that protein he's not consistent. And we don't know why yet. I'm sure people are researching it. But he's inconsistent with how many hydrogens he'll pull through at a time for each electron that goes through him. So he throws off, he's the one that creates the range. But again, you're not responsible for that. Just one glucose will give you about 30. You would never have an answer where it was like 32 or 36. Okay, it would be like very obvious, your alternative answers. But you could be asked how many ATP would be produced from 4 glucoses. How could you figure that out?
4 times 30.
Yep. Yeah, so again, just paying attention to detail with that, okay? You're not typically, in any biology class that I know of, you're not typically asked about 1-pyruvate. It's usually, if it's a big question looking at overall all of cellular respiration, it would be asking about glucose, and you would just have to pay attention to the number of glucoses and remembering that your base is between like 30 and 36.
I didn't get this part of
Okay.
You just like wrote down the equation. I just didn't see the number, but it's 6 glucose are used throughout the thing, right?
No, it's actually 1 glucose.
Oh, okay.
Yes.
It does a lot of stuff.
Yes. So it's 1 glucose, but then it's 6 of everything else except for the ATP.
Mm-hmm.
Hold it, it goes up bigger. I wrote too big on that one.
But 1 glucose. Okay, wait, never mind. I answered my own question.
Are you sure?
Yeah, I was just gonna say 1 glucose makes 2 pyruvate, but I looked at the thing.
Yes.
Okay.
Yes. Very good.
I have 2 questions left.
Yes, ma'am.
And most new stuff comes to me.
Yeah.
You don't need to know about the I don't even know how to say them. The like UBIQU.
Ubiquinone? No. So that's a specific protein complex within the process. And the only specific names that you need are ATP synthase and photosystems. Okay. Those are the only specific ones.
That's nice.
Yeah.
And then okay, the anaerobic respiration goes through glycolysis,
Yes.
and then does it go through anything else?
Yes.
Because
Then it
fermentation
does.
is just glycolysis.
No. Okay. I'm glad you asked that. All right. So glycolysis happens regardless. Okay. Anaerobic respiration, oh gosh, I can't spell, is glycolysis plus whichever type of fermentation, like if we're in a yeast cell, then it would be alcohol fermentation. If we're in a muscle cell, then it would be lactic acid fermentation. But we are going to do glycolysis first, no matter what. Whether we have oxygen or not, we're doing glycolysis. It's just, can that NADH that we just made from glycolysis, this guy over here, can this go to the electron transport chain? It can only go to the electron transport chain if we have oxygen. If we don't have oxygen, then none of these proteins that break down the NADH and the FADH2 will work, because they know there's no oxygen to take those electrons at the end. So instead, you'll do this fermentation and you'll make a teeny tiny bit more ATP. So you get like 4 ATP total from glycolysis plus fermentation.
Okay.
Also, like in the textbook, like it has all of like it has the pyruvic acid plus NADH, like you need that for like lactic acid. Do we need to know like the products and the whatever for fermentation as well?
So you need to know that in lactic acid fermentation that you get lactic acid and ATP, okay? But in alcoholic fermentation, you're going to get alcohol, carbon dioxide, and ATP. Oh, that's really messy. I am sorry. My handwriting is atrocious today.
Okay, thank you. So that's all we needed, because I know in alcohol fermentation it had all this other stuff.
So
Like-
it's including a lot of the NADH, and then that breaks down to NAD plus, and then the electrons. You can get rid of that middle part. You really just need to know, what do we start with and what do we end with? And it's really all about what we end with, because what we start with is from glycolysis.
Okay, thank you.
Yep.
So that's it with anaerobic respiration.
Pretty much, yeah.
Okay.
Yep. Lactic acid happens in muscle cells, so mostly mammals. And then alcoholic respiration, it will happen in yeast cells, a lot of bacteria cells, that kind of, yeah.
Is there anything else you just think we need to know?
I think you just need to, I need you to take a brain break, all of you. You need to take a brain break.
I
Okay?
will.
All right. And then look at your learning objectives. Like I said, they're a lot more general than, like, yeah. So look at those. I apologize for chapters 10 and 11 not being open before this. But yeah, no, I stuck really closely to the learning objectives when I redid the test yesterday. And I think it's a very fair test. I think it is still a little, it's at the appropriate level of challenging, but I definitely think that it's not impossible. Like if anybody from the first year would have said that this test was absolutely impossible, because that year the test was kind of impossible. But it's not anymore.
Yay!
Thank God.
You know, I'm
Thank
looking
you.
more of what's, you know, what is appropriate and what's not at this particular level. It's hard for me to figure out what's going on.
It's hard.
Yeah. So it is hard, but I think it's more doable. You guys are definitely putting in a lot of effort, which is good. So I think that you will be successful if you take care of yourselves today.
Okay, well, that makes me
Right.
feel a lot better.
Good, good. And I'll still be around. I probably won't be answering messages past 9 o'clock. We're going to try and go to bed early tonight. So, but feel free to message until then. And even if you message after, I'll be at school, you know, at my crack of dawn, 7:30. So I can message you back in the morning. Okay. But get good sleep, take care of yourselves. Don't stress too much. You'll melt your brain. You know, you guys should know by now that I take care of you guys. So, you know.
I think my brain's already melting.
Right. So
It's
see,
already done. It's
you
full.
need a break. You need a break.
It's full.
Maybe take an hour break, Emma.
Probably. I probably need that.
All right, guys. So I'm going to call it. Let's go. Let's all have a brain break and rest and relax. I'm going to get this up and on. I have graded all your labs. Look through those. Look at my comments. They were pretty amazing for the most part across the board for everything. So I'm really proud of you guys. But look over those for your lab-based questions too. Make sure you understand big concepts from those labs.
Okay. Thank you so much.
You're welcome.
This really helped. Thank you.
Good. I'm so glad.
See you tomorrow.
See you tomorrow.
Thank you, Miss Shealy.
All right.